Welcome. In this video we'll walk through the 10x genomics chromium single-cell family, what the platform is, the assays it offers, and how to choose between them. If you've ever wondered whether you need Flex, Universal, or an Epi kit, this is for you. At its core, Chromium is a partitioning platform. It takes thousands of single cells or nuclei, gives each one a unique barcode, and builds sequencer-ready libraries. The clever part is that one instrument and one basic workflow powers a whole family of assays. Here's how that works. You mix your cells or nuclei with barcoded gel beads. Microfluidics then partition them into thousands of tiny droplets, called GAMs, in oil. Inside each droplet, every molecule from that cell is tagged with the cell's barcode. Finally, everything is pooled into a library, sequenced, and the reads are mapped back to their cell of origin using the barcode. The family splits into three areas: gene expression, covered by Flex and Universal; epigenomics, covered by the Epi ATAC and Epi Multiome kits; and a set of add-ons like protein, immune repertoire, and CRISPR. Crucially, they all share the same partitioning workflow. For gene expression, the first decision is between two chemistries. Flex is probe-based and covers protein-coding genes. Universal is reverse transcription-based and covers the whole transcriptome. Your sample type and how much of the transcriptome you need usually decide between them. Let's start with Flex. Because it uses probes against protein-coding genes, it's remarkably robust on difficult samples, and it's built to multiplex at scale. It handles fixed and FFPE material, offers massive multiplexing, up to 384 samples in a 96-well format, and lets you run partial plates. The newest version, Flex Apex, adds plate-based automation. That scale is the headline. With plate-based multiplexing, Flex becomes a cohort engine, up to 384 samples per channel and millions of cells from a single chip. That makes it ideal for large, batch-sensitive studies where consistency across many samples really matters. So, when should you reach for Flex? When your samples are fixed, FFPE or archival. When you're profiling whole blood. When you're running large, multiplexed cohorts. When protein coding coverage is enough. And when minimizing batch effects across many samples is a priority. Now the other chemistry, Universal, available in 3' and 5' versions. Universal uses reverse transcription to capture the whole transcriptome, and it comes with the broadest multi-omic options and the widest species compatibility. It also enables de novo discovery of isoforms and long non-coding RNAs, and gives the highest cell recovery for low-input samples. Within Universal, there's a second choice, 3' or 5'. 3' captures the 3' end of transcripts and is the workhorse for gene expression atlasing. 5' captures the 5' end, and importantly, it enables immune repertoire sequencing, TCR and BCR, along with antigen specificity. So if you care about the immune system, go 5'. When should you reach for Universal? When you need whole transcriptome coverage or discovery. When you work in a non-standard species. When you're profiling the immune repertoire using 5'. When you have low input or precious samples. And when you plan to add long reads for isoforms. Putting the two chemistries side by side, Flex is probe-based and protein coding, Universal is reverse transcription-based and whole transcriptome. Flex shines on FFPE and whole blood, with up to 384 plex. Universal is species agnostic, works on fresh and frozen samples, and adds immune and chromatin options. Different strengths, different questions. Beyond gene expression, feature barcode technology lets you layer extra measurements onto the very same cells. You can add protein, both surface and intracellular. You can run pooled CRISPR screens with a single cell readout. And with 5', you can add paired immune receptor sequencing. That immune capability is worth highlighting. With 5', you can recover the paired VDJ repertoire, both TCR and BCR. You can measure antigen specificity, what each receptor actually recognizes. And you can phenotype the same cells with surface protein. Together, that's clonality, specificity, and phenotype in a single experiment. Switching to the epigenome, the Epi kits profile chromatin accessibility from nuclei, resolving cell types and states from epigenetics rather than expression. You can run up to 80,000 nuclei per chip, and there's a dedicated nuclei isolation kit to make sample prep reliable. There are two options, Epi ATAC and Epi Multiome. Epi ATAC uses transposase chemistry to map which regulatory DNA is open in each nucleus. From that open chromatin signal alone, you can cluster cells, define states, and start to read out which transcription factors are active, all from the epigenome. Epi Multiome goes a step further. It measures 3' gene expression and chromatin accessibility in the very same nucleus. Because both come from one cell, you can connect a regulatory element to the gene it controls and begin to reconstruct gene regulatory networks. This is 𝗬𝗃 when regulation, not just expression, is the question. One more extension, read length. Chromium libraries are short-read by default, but universal assays can be paired with long-read sequencing. That lets you resolve alternative isoforms and splicing at single-cell resolution, going beyond a simple 3' tag, all on the same chromium partitions. Of course, generating data is only half the story. CellRanger takes your raw reads and produces cell-by-feature matrices, handling barcode filtering and alignment, with pipelines for gene expression, immune repertoire, and feature barcodes. Loop Browser gives you point-and-click exploration, and everything scales through the cloud and community tools like 𝗬𝗏 and 𝗬𝗵𝘃. And single cell doesn't have to stand alone. Chromium tells you which cell types exist and what they do. Pairing it with spatial platforms, 𝗁𝗍𝗄 for whole transcriptome spatial and xenium for high resolution in situ, places those cell types back into the tissue for a complete picture. So, how do you choose? If your samples are fixed, FFPE, or you're running big cohorts, use Flex. For whole transcriptome work in any species, universal 3'. For immune repertoire, universal 5'. For chromatin accessibility, Epi ATAC. For regulation and expression from the same nucleus, Epi Multiome. And for isoforms, pair universal with long reads. A quick practical note. Match the assay to your material. Chromium supports a wide range of inputs, cells and nuclei, fresh, frozen, or flow-sorted, fixed and FFPE tissue, organoids, and blood, typically around 20,000 cells or nuclei per sample. Your sample type is often the first thing that narrows the choice. To summarize, Chromium is one platform with many assays, all built on GEM partitioning. Flex is for fixed samples and scale. Universal is for whole transcriptome and any species, with 5' adding immune profiling. The Epi kits give you regulation. Add-ons, long read, and spatial extend it further. Match the assay to your sample and your question, and that's the whole decision. Thanks for watching. Visit 𝗪𝗶𝗴𝗴𝗏academy.com to learn more.